Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us, the 136th seminar, and I call it a Serendipity Collective Day. I came back to San Diego after my 18 days of trips to Korea, Japan, Denmark, Sweden to give multiple invited talks and attend multiple conferences, and unfortunately my dad's funeral. Thank many of my friends and colleagues for your kind words and help during this difficult time for me. I think simple kind words or empathy are two of the most important things that make this world a better place to live in, of course, although many sad things such as wars, discrimination, harassment, and hunger still exist in this world after, this is still 2024, very advanced era, but still we have those kind of problems. But I still have very optimistic view of the world because of kind people in the world. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Kev D'Silva. He is at Ideateplus and the CEO and the co-founder of the Serendipity Collective. In the recent decades, innovative or even disruptive research has decreased due to the rewarding system based on the short-term outcomes and incremental progress, as the recent Nature paper discussed. CAP is the strong supporter for innovation, inviting and investing on innovators all over the world and crazy ideas to change the research culture or technology culture in the world. I was invited in 2020, I believe, to brainstorm and compete with other innovators, also collaborate with other innovators in Berlin, Germany, thanks to his vision and support. And I really, appreciate, his effort to make some meaningful change in this community. Kev, thanks for your great idea and vision and contribution to the society by building innovators community, and the virtual podium is all yours now. And thank you again.
Thanks, Ty. And thanks for welcoming me to the 136th conference for SynBYSS as well. Let me just share my screen firstly, and I'll just take you through a brief presentation. So as Ty said, I'm the co-founder of the Serendipity Collective. If you want to learn a little bit about me or the Serendipity Collective, please click onto these, and then you will get access to either of those sites. But just in general, I have been working for corporates for about 20 years in my career. I've been working in strategy, IT, and also global procurement. And then for the last 10 years, I got really passionate about innovation, and that's how I came about to the Serendipity Collective, as well as Ideateplus, which is a Swiss innovation consultancy focused on bringing like a rapid innovation process to corporates, basically. So let me just go through. To start off with, on the Serendipity Collective, we always say, a picture tells a 1,000 stories. So I just want to run through a video in terms of what we did, not in Berlin last year, which Ty spoke about, but also in Tallinn in Estonia this year.
Welcome to the 2024 Serendipity Collective, a platform where a myriad of disciplines converge to share their dreams for the future of science, society, and humanity. Imagine this. It's the year 2100, and humans have built habitats on Mars using self-repairing materials and bionic cells. We've tailored precision foods to individual needs and created digital twins for personalized healthcare. Superbugs recycle waste into valuable resources, and plant cells have been modified for deep space environments. This vision of the future is closer than you think, and it all starts here at the Serendipity Collective. In May 2024, in a transformed factory compound on the outskirts of Tallinn, Estonia, the Serendipity Collective brought together a unique community of innovators, scientists, engineers, artists, and writers.
We want you to intermingle, we want you to learn from one another and really to help drive the science forward.
For 2 days, participants engaged in talks, workshops, and collaborative sessions to rethink, design, and co-develop groundbreaking ideas.
A lot of times in fundamental research and science in general, you see a lot of hard conversations about the deep science aspect of it, and everybody gets very tunneled and very siloed. The artist really brought out the creativity in the scientist, right? And what this, I thought, did was take a lot of the individuals and force them out of that comfort zone when they're a mile deep in that hole and bring them back up to the surface and completely change the whole way they look at that problem.
Big ideas define the future. By providing a platform for these ideas to grow and thrive, the Serendipity Collective makes a world of difference. Our participants' work today will inspire the next generation of creators, contributing to the broader tapestry of humanity.
I'm really sure that we'll use these tools to be better scientists, better innovators, better persons. So I think it's a really perfect event.
To have the opportunity to feel free to think whatever I want to think without any barrier is amazing to me.
The future starts here. Join us in creating a better world. For more information, visit our website at serendipitycollective.com.
So yeah, it was another great event in 2024. So that was in May 2024, and basically it was a little bit different to Berlin. So we had around 40 scientists come. So we had 9 teams, but this time we invited 10 artists as well, international artists, and we kind of asked them to fuse together with the scientists and engineers and kind of forced it a little bit compared to Berlin. And you know, the outcomes were sensational, to be honest. And basically, you know, it was the same platform of people coming together in a safe place, feeling, you know, not judged. No idea was a bad idea. And then ideas were slowly adapting and then, you know, forming along with the teams as well. So we chose 2 ideas to fund. And then, however, what we saw is a lot of collaboration after the event between some of the artists and the teams as well. And that collaboration kept going. Also, we are trying to reach out to more governments as well. So in Berlin, we were with the German government and the US Navy. And in Tallinn, in Estonia, we also had the Estonian government, we had the Indian government, we had the Singaporean government as well. And we're looking to extend that further in terms of our vision of the future. Like you would have heard in the video, you know, Serendipity Collective is a lot about the fusion of art, science, and nature as well. And our role as, you know, in the future of humanity. So it's a little bit different to just doing things for humanity's sake itself, but actually trying to consider what our role would be for a truly equitable society in future as well. A lot of you young stars, you know, up and comers and stuff would probably know about this diagram. It comes from the European Commission. And basically, you know, it talks about what happens to funding as you move along the TRL levels, right? From basic research to implementation. And a lot of you also would be aware of this funding valley of death, basically. So public funding, including governments, academia, universities and stuff, they're happy to fund early stage or pre-seed. And then slowly that funding goes down, but there is this gap before, you know, private funding sees the benefits, they actually want to see the profit, and then they actually rise. So this funding valley of death is something, it's the main reason why we developed the Serendipity Collective in the first place. And we wanted young scientists and young engineers, you know, to have a better prospect of their ideas and to stick to their ideas and not just think about ideas in order to publish papers, but then, you know, actually look at how to get to market with those ideas as well. So if you see now, you know, the Serendipity Collective Ventures will co-invest in actually accelerating those ideas to market. And then we will probably look to exit around a TRL4 and around a TRL5 for when the idea goes into development. At that stage, you know, it's quite a busy ecosystem of VCs or potentially even some governments who are willing to take on these ideas through to implementation. But our sort of sweet spot is in that early phase, and we're trying to address this funding valley of death. We're trying to do that by having government sponsorship for what we call global challenges, which you saw, you know, the output of one of those challenges just in that video. And then after the challenge, we're also now having these governments that we're linking up with and telling them the story of the Serendipity Collective in order to actually have some incubation as well of the startups and the researchers that we actually bring along. The advantage is we get to know these researchers, as we did Thai as well. So it's not just taking startups off the street anymore. It's actually forming those startups from individuals, right? And actually fusing them with different disciplines and just having a better idea and a better prospect of where they're going with their idea in future. So that's why we have, you know, the Serendipity Collective Ventures. That's why it would be important. And that's why it's a little bit different to what's actually happening today. Now we look at, well, what's the value that we bring, right? And we think it's a value for everyone. Firstly, the startups, we're obviously mitigating that funding valley of death, that funding gap from TRL 1 to 5. And most importantly, we're getting young people or older people, because we're looking at, you know, also professors, we're also looking at doctorates and stuff, but we're just getting them to focus on the science. Don't try and be a CEO. Don't try and think about the funding. We'll take that sort of off you, but just focus on the science, focus on your idea, focus on, you know, bringing that hypothesis of yours to life. Also, you know, we're allowing access to expert resources from government organizations and also within the Serendipity Collective ecosystem as well. And the third thing is we're giving them credible validation by having like a government incubation, as we said earlier, and a path to scale up. It's that anticipation or that trajectory of the future that, you know, young scientists find really interesting as well compared to what they have today. So secondly, looking at the governments, you know, it gives these governments access to novel ideas and novel people as well, and novel teams. We're all about, you know, having those teams fuse. It also gives them valid use cases for their incubators as well. So a lot of governments have local kind of incubators, and those incubators can include, you know, certain infrastructure. It can include certain consumables like lab equipment, let's say, or some hardware. And then it also obviously has human capital. So when you talk about countries like Serbia, who is another government we're talking to at the moment, India, they've actually got a very high potential in terms of human capital. and in the English speaking language, and they would like to actually grow that that knowledge within the country as well. And the reason why is the governments have a different objective. It's really about economic growth in their particular countries, and they do that by thinking about ideas, having access to new ideas, and then eventually commercializing those ideas in their country as well. So that's what we're trying to bring to the startups. In doing that, then, the investors, what we're promising them is like a 15 to 20% ROI. We're also giving them a credible validation signal in terms of de-risking through the government involvement. And, there's potential for 10x or even plus growth from about what we're saying, about 10% of the portfolio as well. For us as in the Serendipity Collective, obviously, the key objective is to have this platform of diverse disciplines in order to think 10, 20, 30 years in the future and actually nurture our young scientists as well and give them a path whereby normal academia doesn't actually allow them. And then, eventually grow these little portfolios into multiple kind of funds through the limited partners as well as the success grows. So you can see there's sort of value for everyone in the ecosystem, and that's what we're trying to aim for. So what is the process, you may say? Because, when we have a challenge, let's say we start at the bottom here, and let's just assume it's the 1st of February. Then we call out to you guys to have some sort of crazy ideas, depending on certain topic areas, which I'll talk about in a second. When you actually populate those crazy ideas onto our platform, what we do is we also kind of matchmake skill sets that you need for particular ideas. So if you're a biologist, you may need, I don't know, someone with nanotechnology experience as well. So there is that matchmaking on the platform, and it gives you an opportunity to build your team as well in a virtual state, right? So what we do is we crowdsource the solution basically and form the teams. Then after that, what we do is we have an evaluation event whereby the government sponsors pick out the most promising ideas and then say, well, we will bring about 10 of them to an in-person pitch event, right? And in that pitch event, as Ty has also experienced in Berlin, we kind of try and design the solution a little bit together with all the other teammates. And then we actually pitch your idea. And then we actually propose up to 50,000 USD from each of the governments for the next 3 to 6 months. So then basically you go through, this kind of sprint with, within that 3 or 6 months, whereby you actually create a list of tasks, which are based on the hypothesis that you want to prove. And then you actually go through the typical things that you would in a TRL2 stage in order to further develop your concept. And so basically after that, we have like a progress stage whereby you come and tell us what you have done. And basically then, before the end of that stage, we actually also look for potential future funding as well. And then, get you associated either with the Serendipity Collective or in our case now in terms of government incubators as well for people who are interested in your science or scientific idea. These are just some of the numbers. I won't go through it too much, but you'll see it when you look at the video, and you can look at it afterwards. But we're just starting. So we started a little less than a year and a half ago, and already we can see we're solving a real problem when it comes to researchers, and we will be having impact as well. For example, one of our ideas recently won 3 million funding with the European Innovation Council as well. So you can see that the successes are kind of happening, and they're really valid kind of ideas as well that we're choosing. If you want to have a look at the challenge for December 24, that's already started. So basically, I'll just talk a little bit about, these are just some of the topic areas that we're looking at. Like we said, one of them is around longevity, regeneration, and advanced biomedical technologies. What we're looking at there is around gene regulation and cell function as well. So go and have a look at the QR code and have a look at the platform. You'll just have to quickly register on the platform in order to allow for the matchmaking to kind of commence, but it's pretty easy. And then we've just opened up that challenge, and it runs until the 27th of October, by which stage then we will down select some of the ideas for the December in-person event. We're also looking at things like biosustainability, energy storage and regeneration, and then also e-waste as well. So that's it. I've finished. I probably went over a little bit over 5 minutes, Ty, but we welcome you all to unleash your genius and join us in the Serendipity Collective, and I look forward to answering your questions later as well. Thank you.
Fantastic. That's amazing. So you're having another event soon. So I mean, I encourage all the people with crazy ideas just apply this time again. Okay. Thank you so much. Now, the main speaker of today with a longer introduction, Dr. Manish Kushwaha is a principal investigator at Micalis Institute at the University of Paris-Saclay. His lab applies experimental and theoretical methods to the design of biological systems across scales in cell-free expression systems in bacterial cells of diverse species and also in bacterial co-cultures. He is originally from India, where he did his early education. He moved to the UK for his PhD at Oxford in the field of molecular parasitology, where he studied chromatin remodeling in African trypanosomes, followed by a short postdoc at Imperial College in the same lab. After that, he transitioned to synthetic biology by moving to the lab of Howard Salis at Penn State. Of course, he was the pioneer speaker many months ago. In his lab, Manish worked on the portable expression system called UBER, Universal Bacterial Expression Resource. What a clever name, I would say. He did a second postdoc in the lab of Alfonso Yaramillo at Warwick at the time. Also, he was also pioneer speaker in this seminar series, where he worked on several RNA-based circuits and was introduced to phase engineering. Since 2017, he has been working at the institute in Paris, which is a part of the INRAE, one of the most famous French national labs for agriculture, food, and environment. Manish, thanks so much for your time today. Please take it away, all yours now. Thank you.
Thank you, Jacek. Are you able to see my slide?
Yes, clearly.
Excellent. Well, I'll start then. Well, thank you again, Jacek, for this invitation. It's really quite an honor to be invited to this SynBYSS seminar series, which as I was telling you earlier, I follow quite regularly. So I know that, in fact, multicellular circuits is not new to this series. Many people have spoken about genetic circuits in bacteria, in single bacteria, and in multicellular circuits, most recently, I think, by Lauren Andrews. So this audience is very familiar with building circuits with bacteria. Before I go into the science of it, I want to quickly just say where I'm located. So I'm located at the Micalis Institute. This is, in the French system, we say it's a mixed research unit, because this is, in fact, a collaboration between two national labs, two research organizations, so INRAE and AgroParisTech, as well as the University of Paris-Saclay. It's located in the southern suburb of Paris. It's a place called Jouy-en-Josas, not very far from the beautiful Palace of Versailles. And in fact, in our institution at Micalis, there are about, I would say, more than 20 teams. So it's quite a decent sized microbiology lab with 3 main themes running through these different teams on essentially host pathogen interactions, on microbiota of food and intestinal systems, and synthetic and systems biology. And no surprises for guessing that I am a part of the third one of these themes, the synthetic and systems biology stream. So just to also give you a little sense of what kind of a campus it is, it's a very green sort of rural campus away from the busy Paris life. And we have lots of animals on the site, and we have quite a lot of green patch of campus. So it's really quite a nice setting for research. Going on to the engineering of these multicellular circuits themselves, we know that engineering cellular machinery has many advantages. Definitely in the field of synthetic biology, this is almost a truism. Tracing back the engineering of cellular machinery to the earliest, recombinant DNA technology, the production of insulin early in the 1980s, to now where many different kinds of circuits can be built with biological technology. Here I have an example of circuits, in fact, built by Tae-Suk, published in 2012, which sends, in fact, many different small molecules and give you an AND gate response in this case. On the right, we have some circuits, in fact, that came from our team, where we multiplex the sensing of many different metabolites in cell-free systems. So you can see that, in fact, a lot of work has gone into building genetic circuits for information processing in synthetic biology. And a lot of it still remains a very fundamental exercise, being able to build these circuits. But of course, the larger aims are that then you can use these circuits to do something useful, something more applicable. And more recently, there has been a trend towards incorporating circuits in real-world applications. For example, building smart systems like this example from Taldanino's lab, where 3 different inputs can be sensed in order for these tumor sensing bacteria to be able to target the tumor in a mouse model, right? So we know that synthetic circuits can be very, very useful, and they have many applications for us. But there is a problem that a lot of synthetic biologists, particularly those interested in information processing genetic circuits, are aware of, and this is about the expenses of maintaining and running this machinery. So whenever you put all these heterologous genes that are meant to process information inside the cell, it leads to a load on the cell. And this, I really like this one figure from the Cello paper of 2016, where the author sort of mapped the success rate of the different circuits against the number of transcription factors that were in the cell, that were added in the cell, right? And they find that, in fact, as the number of transcription factors go up, the chance of failure also increases. And part of this is due to crosstalk inside the cell, but a big part of it is also the load on the circuit. And in fact, the fact that genetic circuits are quite burdensome for the cell had been figured out much earlier, and in fact, solutions for this had been proposed. So already in 2011, we saw 2 very interesting papers published in the same issue of Nature, one by the Voigt lab, the Tamser et al. Paper, where a bigger complex circuit was in fact broken down into simpler circuits and installed in different cells, where these cells can then communicate through small molecules and therefore recapitulate the bigger circuit. Similar work was done using yeast cells and hormone molecules as communication channels in Regaud et al. In Ricard Soleil's team, where in fact these yeast cells were able to talk to each other using pheromone molecules and therefore build a bigger circuit. So of course, the advantage of having multicellular circuits is that you have a division of labor. Not all the machinery is in a single cell, so your cells are less stressed and you can have bigger circuits. It also minimizes crosstalk. So if there are any cross-talking agents, they will have to cross the cellular barrier in order to go to the other cell, and this, of course, is less likely to happen in multicellular circuits. And of course, you can create specialized subfunctions, meaning each one of these cells then represents a smaller subfunction which is unique, and in fact, you can mix these different cells to sometimes generate newer functions as well. Right. So we know that multicellular circuits can be quite useful in many of these regards. However, one of the challenges with multicellular circuits, as you may have already guessed from the previous slide, is that you need these external wires to connect these different sub-circuits. And work has happened over the last 10 years, I would say, to develop many different kinds of external wires to be able to connect circuits. So people have repurposed secondary metabolites, about 6 orthogonal versions of them, to be able to connect different cells, or engineered synthetic receptor ligand pairs in order for mammalian cells to talk to each other in a contact-dependent manner. Or very recently, in fact, Tom de Grief's lab had this paper where they used synthetic coil-coil ligands to, in fact, engineer cell-to-cell communication. In this case, these ligands are diffusible, so they can be produced by one cell and then received by the other in order to communicate. So there is a growing number of orthogonal external wires that exist, but in fact, making new orthogonal signals remains quite challenging. It is a lot of work to generate a new signal, make sure that it doesn't crosstalk with another existing signal, and so on. So being able to build unique external signals is quite useful for building bigger circuits. And in this regard, in fact, we know that cells don't interact in nature just by small molecules or by contact. They can use even nucleic acid molecules to interact with each other and, in fact, have a bigger information exchange between them. And in fact, this quote from Jean and Peter Medawar from the 1970s, who were both pathologists and virologists, where they say, a virus is simply a piece of bad news wrapped up in protein. So here I highlighted the word news because, in fact, news is a message, it's information. And what a virus is doing is transmitting this nucleic acid information from one cell to another. And this idea, well, we're all aware of this because we've recently gone through a global pandemic. It means that this message can be transmitted from one cell to another into whole populations. And in fact, each one of these members in the population becomes a processing machinery for the virus, whereby it can, it can not only propagate itself, but also, if engineered correctly, make certain decisions. And this idea, of course, is not entirely new to SynBYSS. Almost 12 years ago, the lab of Drew Endy, in fact, already identified this and tried to use M13 bacteriophage for developing cell-to-cell signaling in this paper. And in fact, M13 is quite a good phage to do this because it's non-lytic phage, so it doesn't lyse cells when it produces phage particles. It can simply secrete out of the cell, go infect another cell, and thereby transmit this message from the sender to the receiver. Because the message is in fact a DNA molecule, it's a very programmable signal, and it achieves what the authors defined as message channel decoupling. And this is quite an interesting idea in that for all the small molecule-based communication, you always need a specific machinery to be able to sense this molecule. So if you have 4 different HSL signals, these quorum molecules, you need 4 different transcription factors. And therefore, each of the messages is being transmitted, as it were, in a different channel. But when you have DNA message being transmitted, it's the same phage machinery that can be used. And so within the same channel, multiple different kinds of messages can be propagated. And I think this is quite a nice idea. In general, it applies to all kinds of DNA messaging, not just phage messaging. It could apply to conjugation-based messages. It could apply through package vesicles that carry any nucleic acid and so on. Right now, because the M13 phage is not a lytic phage, it's a filamentous phage, it secretes itself, infects another cell, it has all these different advantages. We already discussed that it results in message channel decoupling, the signal is executable, it has a large packaging capacity because it does not need a well-defined capsid, it can grow in size depending on the amount of DNA that's packaged. And already it is used in a lot of different technologies. So phage display technology, in a lot of nanotechnology, in the evolution systems of PACE and PACEMED. So in fact, the biology of this phage is very well known, and in fact, it can be quite easily adapted to cell-to-cell signaling, as we've already seen. And in fact, it is quite simple. It's deceptively simple, I would say. All the phage machinery goes in a plasmid called the helper, marked here by an H, and the message to be transmitted is in fact a phage-mid molecule, which is a plasmid with a phage packaging signal. So the helper machinery just takes this molecule, packages it as a phage, puts it out there. This one infects receiver cells, and voila, you can see that the message has been transmitted, right? Now, what do we do with this is, of course, depends on how we design this DNA molecule. So in our case, we were interested in building two different kinds of phage-mids. So we have a phage-mid, and in both of these cases, what we call the minus GP3 phage-mid and the plus GP3 phage-mid, all the phage machinery is already in the sender cell. The only difference is where the GP3 gene, which is in fact an essential minor code protein of the M13 bacteriophage, is located. And it's located on the helper in the case of the minus GP3 sender, but in the case of the plus GP3 sender, it's in fact located on the phage-mid itself. And in fact, we wanted to do this because we wanted to test what works better for which application. And I won't go into that into great detail, but simply put, minus GP3 phages are useful for delivery of payload, but they cannot resecrete. If you have plus GP3 phages, you could design the receivers to be able to resecrete the received phage. And it was that intention we built these two different variants. And in fact, we tested what the secretion rates of these were, and we found that there were, in fact, significant differences in the way the two phages secreted. The plus GP3 senders grew really fast with a very little lag phase. The minus GP3 ones had a longer lag phase, but in the end, they reached similar ODs at endpoint. But there's dramatic differences in the secretion rates of the two phages. So the minus GP3 phages, you get a lot of phages, by the end of the 15 hours, but the plus GP3 phages, the production rate is very, very low. And when we calculate the secretion rate per cell, in fact, we find that the minus GP3 phages produce around 0.7 phages per cell per minute, but this number is almost 1,000 times lower for the plus GP3 phages. Now, there is a direct relationship between the secretion rate and the growth rate. And when we plot the growth rate against the secretion rate per cell, we find that, in fact, the higher the growth rate, the more the secretion rate. And we're not the first one to report this. This has been seen before with natural M13 phages. But we find that, in fact, there seems to be a plateau beyond a certain growth rate, which is about 0.4 per hour. You don't get any additional per cell secretion rate because probably at this point, other components have become limiting for phage secretion. Now, we were interested in asking, so just like we were interested in asking how the growth phase of the cells, in fact, influence the secretion rate, we were also interested in knowing how the growth phase of the receiver cells affects the infection of these receiver cells. So in order to do this, we devised a little experiment where we grow these cells to different growth phases, but then in normal batch culture, what you get is the growth phase and the density of cells change at the same time. So it's harder to dissect where the effect, if any, would come from when you try to infect these cells. So what we did was we grew them to different growth phases, but then resuspended them to the same density and then infected these cells with the phages to identify that, in fact, the later growth phases are more receptive to phages, and you get higher infection rates per cell in the later phases of growth than in the early ones. And this told us then that, in fact, it's very important to consider the growth physiology of the cells when designing our circuits. Next, we wanted to look at how the infection and selection dynamics proceed in growing receiver cells. So what we did here was really quite simple. We have a microplate, a 96-well plate, where we split this into two segments, each segment having different concentrations of initial starting phages and receiver densities. We incubate them for 1 hour without selection, and then we grow them with selection, with or without kanamycin. The infected cells will resist kanamycin and therefore be able to grow. The sensitive ones are going to die. So by doing this, we can see, I mean, this is completely expected, that the rescue under antibiotic selection is both phage and receiver dose dependent. So the more phages you have, the more rescue you get, the more receivers you have, the quicker rescue you get, in fact, because the chance of the infection occurring, of course, increases when either of these two components is more. But what is more interesting to, in fact, observe here is that when you have high phage concentrations, you almost always get a deterministic behavior. So you get very low coefficient of variation looking at these infected receivers. But as you go down on the number of phages, you start getting this sort of stochastic behavior where the different repeats don't always have similar trajectories. And of course, when you get too low with the phage concentrations, you don't get any infection. And as a result, you have a lower COV again. But what this tells us is that, in fact, you need an optimal sort of number of phages in order to be predictably able to deliver your payload. Otherwise, you get into the stochastic regime where infections may or may not happen when you're using isolated phages. Next, we wanted to ask how this works when you use sender cells directly rather than isolated phages. So we set up a similar plate reader experiment where you have different deletions of senders and receivers that are mixed together, incubated for an hour, and then selected, and different antibiotics that select for either receivers, senders, or infected receivers. And by looking at the growth curves in these different conditions, you can get some idea of what's going on in the sender-receiver system. So we can clearly see that growth rescue under antibiotic selection is again sender and receiver dose dependent. This is a very expected response. The more the senders, the more the phage gets transmitted, and therefore you have more infected receivers in the end. But what is more surprising, in fact, for us was that the fastest infections, although they occur in high sender, high receiver settings, there is an effect of resource competition that you see between senders and receivers, and this tends to shape the communication outcomes. And here I will point you to, very specifically, I'll point you to these receiver populations at the endpoint, and you can see that here, when the initial amount of senders was high, in the end, the OD we get is lower. And this tells us that if we add too many senders, then in fact, in this co-culture, they will take away the resources that are required for receiver growth and will in fact reduce the total number of infected receivers in the end. So this competition between the two cell types is important for these communication outcomes in the end. But what was in fact very promising for us was that when you use sender cells, in all the different settings that we tried, we never get high coefficients of variation. They're always under 25%, which is much lower than what we've seen for isolated phages. So when using senders for infection, variability gets reduced a lot due to this amplification effect that we see. And in fact, what we estimate is that, at least for these minus GP3 phages, if you have any more than 15 phages per mil, you will almost always get deterministic behavior. And you really need to go down very low with the sender numbers in order to go into the stochastic regime, which means that these findings could potentially be used to improve, payload delivery in complex environments, for phage therapy, and so on. Next, we wanted to now use these tools to be able to build some logic gate circuits using the senders and receivers. So here what we have is simply testing a simple NOT gate. It's a CRISPR-I system where the guide RNA is encoded on the phage particle itself. It is delivered into the receiver cells, and there the guide RNA expresses binding to the Cas9 that is already constitutively expressed in the receiver cells and prevents the expression of the GFP by, in fact, binding downstream of that promoter. By having a simple circuit like this, we can look at the rate of phage infection, and we can get some idea of the kinetics of this process, both with isolated phages and senders. And in fact, that's exactly what we did. We ran these cells, we collected these cells at different time points and ran them to a flow cytometer to be able to identify cells that had turned off the GFP. We can see that already by 5 hours, 90% of the cells had reached off state. Now, this is quite comparable to, in fact, the results that Ortiz and Andy had found in the 2012 paper, where they also found that in 5 hours, 92% of their cells were infected by the senders. Now, it's important to remember here that we do not use any antibiotic selection. So here, all of this transmission is happening without any antibiotic selection. We tested this. So here we tested this with the isolated phages, but then we also tested the same with sender cells. And in fact, what we find is that even when the sender cells are, much fewer than the receivers, so mixed in the ratio of 1:4, senders to receivers, you get very high infection rates at 5 hours, so almost 87%, and this number goes up as the sender concentrations go up. If you keep the sender concentrations high and the receiver concentrations change, this doesn't seem to affect the rate of transfer, because already the senders are in excess, so a lot of these phages are being produced. We use this data, in fact, to calculate the transfer frequency, which is the ratio of the infected receivers over the product of senders and total receivers. And in fact, this is quite commonly used So this method is quite commonly used to calculate transfer frequency in conjugation experiments to assess transconjugation. And in fact, what we found was that using phages instead of conjugation gives us almost 5 orders of magnitude higher transfer frequencies. So in a recent paper, in fact, from the Murray lab, they had used conjugation for such DNA transfer. And in fact, in 6 hours, the transconjugation efficiency they found, ours is many times over that at 3 hours. So this tells us that, in fact, phage-based communication is quite efficient compared to, for example, conjugation. Next, we wanted to use this to implement logic gates. And here we have, in fact, the same guide RNA that was used before being transmitted in the form to create this NOT gate. The sender cells produce a phage that expresses the sgRNA1 that then represses the GFP. And what we wanted to test here was, we've already tested that at 4 hours, but we wanted to see whether at 16 hours it would be any better to apply selection for the infection by these phages. And in fact, what we found was that, at least for this NOT gate, applying selection does not seem to improve the outcome. So even without any antibiotics added, simply the sender cells being added to the system, there is a 20-fold repression of the GFP in the receiver cells. If we do apply selection for the phages or just the receiver cells themselves, the outcomes are similar. So this tells us that, in fact, at least when a single guide RNA is being transmitted, selection does not seem to be required. It is quite efficient, and it can proceed quite rapidly. We wanted then to use this to build other circuits. So here we have a yes gate implementation where a guide RNA, so here guide RNA2 is secreted by a different sender, which then in fact blocks the expression of an internal guide RNA that we call guide RNA X, thereby creating a double inverter. And here adding the guide RNA, in fact, increases the expression of the GFP. And we can see that, that we get about 17 to 21-fold activation, depending on whether selection is applied or not. So even here, without selection, we seem to get better activation. With selection, we still get comparable activation, but there is small differences. But one thing to point out here is that in this particular case with the yes gate, with just the pre-incubation at 4 hours, we did not see such a high activation. And we think this is because there is an internal cascade. Well, there could be both. The rates of secretion of the guide RNA of this phage may be different. And also there is a cascade that needs to happen, one guide RNA turning off another, and this probably delays the activation of the yes gate. Next, we wanted to build gates that can take more inputs and not just one. So first, we tested whether this is possible. We built different kinds of senders that produce phages with different antibiotics and replication origins. And we tried to infect a single receiver with them and select them on multiple antibiotics. And we can see that, in fact, multiple senders can infect a single receiver, but the growth rate suffers a lot. And this told us that, in fact, if we want to build, multi-input gates in using these phages, we should apply some kind of a selection. Otherwise, this may be a hard task. Given we already saw that with the yes gate, we did not get good activation at 4 hours without applying selection. But then this leads to a problem. If you apply selection for certain receivers where an input has been added and don't apply it to the others, then it's very hard to compare the fluorescence between them because the growth rates of these cells are going to be very different. So what we did was, in order to build these multi-input gates, we in fact switched our strategy to a dual rail encoding instead of a single rail encoding. And in a single rail encoding, as we've seen before, the presence or absence of the sender represents whether the input is treated as on or 1 or off or 0. But in a dual rail encoding, what we have is, in fact, for every input, in this case, let's say input A, you can have two different kinds of senders that produce either a 0 signal or 1 signal. And therefore, your 0, 1 is, in fact, two different variants of the sender. Similarly, we have two different variants of sender B, so we can get guide RNAs B0 and B1. And in fact, these two guide RNAs, the A0 and B0, are in fact dummy guide RNAs that are non-targeting. So they don't target the promoter of the GFP or the promoters of the internal guide RNAs, and therefore they act as dummies, but you can still select for them because they do have the antibiotic resistance marker on them. So when we did co-incubation and selection, using these sender combinations, we can really nicely see an AND gate behavior. When the right form of the input, which is a 1-1 input, is applied, we get 17-fold activation when we compare it to the mean of the other 3 states. So it's very promising to see that you can have such a 2-input gate. We wanted to expand this further to increase the number of inputs. And so we added another guide RNA binding site downstream of the GFP promoter. And now what we have is, in fact, an AND and NOT gate. So a 3-input gate where 3 of any 6 senders can be co-cultured together and then selected in order to see this AND and NOT behavior, where the output is on only if A and B are on or in state 1, and C input is in state 0. So here we see a 10-fold activation when comparing against the mean of all the other states, which is still quite a high fold activation for a 3-input circuit. We were interested in looking at how this plays out, kinetically. So we looked at the plate reader data to see how quickly these gates had started getting activated. And in fact, as expected, we found that the time to switch really depends on the number of the regulatory steps. So when you have a NOT gate, you get a very quick response, and we define our switch state as being, we define our response time as 20% switch in fluorescence. So if we use that metric, in fact, within 40 minutes, the NOT gate is already beginning to turn off. Sorry, the yes gate is beginning to turn on by 170 minutes. When you have the AND gate, it takes longer, and the AND and NOT gate even takes longer still. And the reason for this is, of course, the cascade of these regulatory circuits, but also there is likely a delay because multiple infections have to happen in order for the circuit to be turned on. Now, the NOT gate activation of 20% here compares quite well, in fact, to what is seen with quorum molecules. So a lot of the experiments with the quorum sensing molecules often use enrichment strategies, sometimes using conditioned media, at other times repeatedly growing cultures in the same media to enrich the numbers. But if you're looking through the literature, in fact, what we find is, depending on the concentration of the molecules added, it can take anywhere from 35 to 100 minutes to reach 20% activation with small molecules, or when using senders, in one study, it took about 150 minutes to activate the circuit by 20%. So this shows us that, in fact, our phage production is actually quite good, almost comparable to small molecules in terms of the rate of activation. In summary, I've shown you that phage-mediated communication system can be coupled with CRISPR-I to generate an intercellular CRISPR interference. Cell physiology affects both phage secretion and infection rates, and resource competition shapes this sender-receiver communication behavior, both in terms of the outcomes, but also in terms of the variability in the outcomes. Several phage signals can be generated quite quickly to make orthogonal messages that can be transmitted through the same channel, as well as phage messaging can be used, as we've demonstrated, to build multicellular circuitry for different kinds of logic gates that take 1, 2, or 3 inputs, as demonstrated. Right now, we are trying to expand this work to build, selective amplification of phage signals, try to get multi-layered circuits going, and try to apply this for DNA payload delivery, possibly in microbiomes or bioreactor settings. This work we recently, in fact, last week, put out as a preprint. So for the audience, if you have any comments on the preprint, they're very welcome. In the end, I would like to thank all the people that were involved in this work. So bulk of this work was done by a PhD student, Abhinav, and postdoc Amit, and also a former PhD student who really helped us set up this a few years ago, master student Corbin Hopper, and our collaborators from ENS Paris-Saclay, Matthias and Thomas. And big thanks to all the members of the BioRetrosynth team and the cellular computing group, as well as my former postdoc advisor, Alfonso, where I learned to use the M13 and really also CRISPR systems. With that, thanks to you all for listening, and I'm happy to take any questions.
Fantastic. I love it, especially your multi-sender system to one receiver system working beautifully, and that's amazing. And also, we recently, my lab developed liposome as a DNA delivery system.
Wow, yes.
I'm always aware of the phage could be the alternative, and then you demonstrate beautifully about the delivery of the DNA message to the bacteria. Let me ask one question, that to make your system much, appealing, or the more applicable to their other kind of application. I mean, at this moment, this is already kind of giving a lot of potential, but if I didn't miss, one thing I want to see is bidirectional communication. So if I remember correctly, or understood correctly, that's currently unidirectional. Am I right?
That is correct. Yes.
So,
absolutely.
But the bidirectional is more complicated because you need to make sender cell and receiver cell doing the both job orthogonally. So could you kind of comment on that point and then elaborate how you're gonna achieve those kind of a little bit actually a very challenging goal? I would say it's not simple, but just tell us what you will do.
Yeah, so I would like to, so I had made a little comment on this slide, which is, so there is a problem with M13 and a lot of phages, in fact, is that they naturally don't want to have super infection. So they don't want that multiple phages of the same type can infect the same host. As a result, many of them have what is called superinfection inhibition. In the case of M13, this superinfection inhibition operates at the level of this gene, the GP3. So if GP3 is already expressed inside the cells, then they will not take any additional phages.
Yeah.
Now,
yeah.
that
That's
is
what I
why all our multi-input circuits, we always use the minus GP3 phages.
Got it.
Now, if we use the minus GP3 phage, the problem is the receiver cell does not have GP3, so it cannot package a phage and send it out, right? And so really the challenge now, this is not, I think, a challenge that's impossible to meet. We're already working on it. We already have some promising leads, is really to be able to express GP3 in a tightly controlled manner. So if you are able to, inducibly express GP3, then you can get this bidirectional communication going. Otherwise, because the very presence of GP3 in the cell prevents further infection, this is very hard to achieve right now.
So, but you are working on that goal right now.
Yes, yes.
Fantastic.
I mean, clues to this already exist. So, like the stringency control PACE from David Liu's lab, they already put the GP3 under inducible expression, and in their system, it's something that is possible for GP3 to be expressed only under specific conditions. And so this is something on those lines we want to explore for this bidirectional communication.
Fantastic, because if you achieve the bidirectional communication, that will be very cool. I mean, also there are multiple applications we may envision, because I mean that kind of giving us huge opportunity and then complex DNA exchange system that kind of basically open the completely new the direction at the end of the day.
Yes.
Okay, let me check whether we have the question from the audience. I don't see, and then I now see the time is already 6:11 Central Time, and then I believe Paris time is already 6 p.m. whatever the dinner time.
6:06, that's right.
Yeah, so probably wrapping up. Let me double check one more time. Q&A. Okay, so no question from the audience right now. So let me close by saying something. So thank you so much all for joining.
Thank you, Jason.
Pardon? Oh,
thank you. Thanks to you. It was a great opportunity to really present here, and it's really an honor. As I told you already, I follow the SynBYSS talk, so it's great to be on them.
Yeah, it's my honor to have you. I mean, the future leaders, of course. So let me close again. Thank you all for joining and staying today. We'll meet again on September 12th, Thursday, the same time, the same Zoom link. We'll have the Dr. Martina Siwek, a regional chief scientist at Office of Naval Research Global. And in fact, I met her during the Serendipity event 2 years ago, I believe. And Professor Jung-Wook Lee at Pohang University of Science and Technology, or simply POSTECH. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I will promote you to panelists who can speak and show your handsome and pretty faces if you wish. I thanks again, and I stop recording. Just give me 1 second.